Good day, this is Professor Will, CBMD PhD. Today is Monday, June 10, 2019. It is 23:41:39 seconds Eastern Standard Time. And this is a continuation of my scientific research. So let's get started. Ultimate Destiny: How Undifferentiated Cells Commit to Their Biological Fate. Date June 6, 2019. Source: Harvard Medical School. Summary: a new study using mouse neural crest tissue reveals how primitive undifferentiated cells choose their adult fate. The findings help explain how some neural crest cells become craniofacial cells, while others end up as nervous system cells. The research can help pinpoint where cells may take the wrong turn and become malignant. The complexity, however, belies the fact that each of the trillions of highly specialized cells start out as a single primordial cell. How do these primitive, undifferentiated cells choose their ultimate destiny? It is a question that has tantalized biologists for centuries. Researchers have uncovered intriguing new clues about the molecular logic of cells that informs their fates. The findings, published June 7 in Science and based on a study of mouse neural crest tissue, show that cells face multiple competing choices on their journey to adulthood and perform a series of binary decisions until they reach a final destination. A progenitor cell could become anything, but how is that choice realized? Our study represents an attempt to define the molecular logic behind cell choice. We believe our findings can help us understand how cells orient themselves toward a particular fate and what might go wrong in the process of cell differentiation. The research reveals that neural crest cells' decision occurs in three phases: activation of competing genetic programs vying for the cell's attention, gradual biasing toward one of these programs, and the cell's ultimate commitment. The researchers caution that at this point, their findings pertain solely to neural crest cells, but the same approach could be explored to understand cell differentiation in other tissues. It remains unclear whether other tissues, organs, and organisms follow a similar mechanism of cell differentiation. Beyond shedding light on a fundamental question in biology, the study results can help eliminate what goes awry in stem cells that take the wrong turn and become malignant, or help inform new techniques for growing artificial neural tissue for medical treatment. We hope our findings can provide a new window into a diversity of neural crest cells and help explain both the normal development of cells that give rise to craniofacial, heart, and sensory tissues, but also some of the pathogenic detours along the way that lead to abnormalities of cell differentiation. Such insights are critical not only for understanding the fundamental biology of cellular differentiation, but possibly for informing therapeutical strategies down the road. A cell's dilemma. The researchers trace the trajectories of primitive cells from mouse neural crest tissue, a collection of cells that arise from the ectoderm of one of the primary germ cell layers formed during embryonic development. These progenitors give rise to a variety of cells, including different nerve cells in the brain, spinal cord, and elsewhere in the body, pigment-producing cells, as well as the cells of the bones, cartilage, and smooth muscles that make up the skull and face. To retrace the decision-making of these primitive cells as they diverge into different areas of specialization, investigators use single-cell sequencing, a technique that allows one to observe the genetic changes in individual cells, one cell at a time. The researchers plotted a cell trajectory in the form of decision-making trees, marked by a series of forks on the road. To determine the sequence of a cell's decision and how it commits to a given fate, the scientists tracked the rate of RNA change in individual cells. RNA changes, measured by shifts in the rate of gene expression and protein production, occur when a cell is starting to carry out marching orders from genes and to transform itself. As genetic programs get activated or silenced, the rate of RNA production changes accordingly. Much to the researchers' surprise, the analysis revealed that competing groups of genes, genetic programs that regulate various cellular functions, nudge cells simultaneously toward different developmental pathways. As cells decide on a path, one genetic program becomes stronger, while the competing ones get weaker, allowing the cell to move toward its chosen path. The analysis shows that cells face a series of binary choices, with each subsequent decision further narrowing the choice of specialization. For example, the analysis demonstrates that the first bifurcation on cell's journey occurs at an intersection when a neural crest cell must choose whether it will become a sensory nerve cell or another type. At the next fork on the road, the nerve cell must decide between becoming a glial cell, a type of cell that supports and shields neurons, or a neuron, and so forth until a final state is reached. The next question the scientists wanted to answer was how a cell steered toward a particular fate. Does the cell slowly start to activate the molecular machinery that pushes it on the right path, or is there something else going on? The findings reveal that individual genes do not bias the cell's choice independently of each other. Instead, whole clusters of genes associated with distinct fates get activated simultaneously and vie for cell's attention. The closer the cell gets to a decision fork, the greater the co-activation of the cell genetic programs, each beckoning the cell in a different direction. A decision between becoming a jawbone cell versus a nerve cell, for example. The observation suggests that a cell makes a choice only after both programs are partly activated, priming the cell for both alternatives before it commits. Once the choice is made, the irrelevant genetic program is silenced. That was rather surprising. We expected to see something simpler like the cell showing earlier preference for one option over another. Instead, we observed that the cell prepares for both options, considers both options, and only then commits to a decision. The findings suggest that a complex long history of conflicting signals gradually prepares a cell for a range of possible outcomes, a journey that ends with distillation of these choices into a single available option. The researchers caution that their findings reveal the internal decision-making of a cell, and how a decision is executed, not the factors that actually guide the ultimate choice. Those factors are likely external signals from a cell surrounding, rather than signals that arise from within. The cell, however, must be primed to respond to the relevant external signals when they arrive. What we see is how the cell prepares for that decision and gears up to respond to one call or another. Something pushes the cell in one direction, but we still do not know what that catalyst is. Veering off the right path. The observations can help scientists understand how cells mature to perform their roles, but just as important, how they may veer off course and begin to divide uncontrollably. The cardinal feature of cancer. For example, the findings can illuminate the processes that underlie diversity in tumor cell populations and resistance to therapy in certain pediatric cancers called neuroblastomas, tumors that arise from immature nerve cells. Several types of cancer originate from the neural crest lineage, including tumors of the peripheral nervous system, some endocrine tumors, and melanoma. Although cell specialization is a tightly controlled process, errors of differentiation can occur, giving rise to malignancy. There has been some indication that neural crest tumors arise from a cell's inability to overcome differentiation forks on the road and it gets stuck. Moving forward, we want to find out at what point a cell breaks away from its intended path and starts to overproliferate. The researchers said that conducting a similar analysis on human neural crest tissue to detail the precise molecular events that occur at these critical junctures, and how cells resolve or fail to resolve them, would be an important next step to understanding the changes in genetic programs that accompany normal and abnormal differentiation. Today's is Professor Will, CBMD PhD. Today's Tuesday, June 11, 2019 at 0048 9 seconds Eastern Standard Time. Continuation of my scientific research. So let's get started here. Translation of genes more complex than expected. Date June 6, 2019. Summary. Researchers have shown that translation of genetic information stored in our DNA is much more complex than previously thought. The discovery was made by developing a type of advanced microscopy that directly visualizes the translation of the genetic code in a living cell. From gene to protein, each cell in our body contains the same DNA, yet different cells, like brain cells or muscle cells, have different functions. The differences in gene function depend on which parts of the genetic information called genes are active in each cell. The genetic information stored in these genes is translated by specialized translation factories called ribosomes. Ribosomes read the genetic code and assemble proteins based on the information stored in this genetic code, analogous to a factory building a machine based on a blueprint. Proteins are the workhorses of our body and perform the functions encoded in our genes. For our cells and organs to function correctly, it is critical that the genetic information stored in our genes is translated accurately to proteins. If the genetic code is translated incorrectly, harmful proteins can be produced, which can lead to neurological diseases such as Huntington's disease. The reading frame of genes. The genetic code is translated in groups of 3 letters, each resembling a word, which is translated into a single part of the protein. If a ribosome starts translating the code at the wrong position, a shift in the 3-letter code can occur. In the case of the genetic code, this phenomenon is called out-of-frame translation. A new method, SunTag and MoonTag. The researchers developed a new method to visualize the decoding of our genetic information in living cells. They were able to label different protein products in different colors and visualize the production of each type of protein using advanced microscopy. Today's is Professor Will, CBMD PhD. Today is Tuesday, June 11, 2019. It is 1:01:21 seconds Eastern Standard Time. And this concludes a brief description of my scientific research. Hope you enjoyed listening. Have a great day.